Hello, everyone. Today, I'll be sharing some common standard questions and answer in the N-level biology syllabus of the Singapore version. I'll be going through 30 questions with answers. These are the standard questions and answers that normally appear in school prelim paper and the GC N-level examination paper. I'm going to use this opportunity to share with students how to answer standard questions. Of course, there are other types of questions like database inference, definitions, application questions that are not the so-called standard questions. However, for GC N-level, there are still questions that are rather standard that appear almost every year. I have two students who are doing normal acad biology. Based on my observations, they have difficulties or challenges to remember certain details. So what I did was, during the lesson, I will give them time to record the information and highlight keywords for them to remember. This will greatly help them to remember and recall the important facts and write them down during the examination. So I'm going to go through standard questions, starting with number 1. Describe how the red blood cell is adapted to carry out its function. So what are the key points to take note? Number 1, it is important to take note that the questions may ask students to provide more than one adaptation. So it depends on the question. If they say 2 adaptations, you gave 2. What if they never specify the number of adaptations? Then you will give according to the marks allocation. But it may not be a 1 mark, 1 point thing. So it could be this. 2 marks question will require at least 2 adaptations. So the answer will be red blood cell contains hemoglobin to bind to oxygen and transport it around the body, which is not evidence in other animal cells. Okay, red blood cell is adapted, meaning what makes it special in order to facilitate in carrying out its function. Number 2, red blood cell has a circular biconcave shape to increase its surface area to volume ratio to enable faster rate of oxygen uptake and release. Red blood cell does not have nucleus to allow the cell to store more hemoglobin. Red blood cell has a thin and flexible membrane to allow it to squeeze through the narrow blood capillaries. So I will expect students to remember all these 4 adaptations in the syllabus, not just for structured question, but also it may come out in the database inference questions or MCQ. Okay, let's move on to the second question. Xylem vessel. Okay, the order of the question followed the order of the topic in the textbook lah. So that's what I actually did. Xylem vessel has a long, hollow, narrow tube extending from the roots to the leaf with no cross wall and protoplasm to enable no obstruction to water flow in the lumen. So these are the important key points. We need to mention that xylem vessel is long, hollow, and narrow, starting from the roots to the leaf. It is important to recognize that there's no cross wall or protoplasm to prevent the flow of water in the lumen. Number 2, Xylem vessel is thickened with lignin to provide structural support and prevent collapse of the xylem vessel. So there are two functions of the xylem. Number one, transport of water with mineral salts. Number two, structural support. And these are the elaborations. For root hair cell, these are the adaptations to carry out its function efficiently. Number one, you need to mention root hair cell has a long and narrow protrusion to increase surface area to volume ratio. This term is very commonly described in the biology syllabus. You will use it very often. This is not the first one and not the last one. So why does it need to increase surface area to volume ratio for? That is to facilitate the absorption of water and mineral salts. Root hair cell has many mitochondria to release energy for active transport, allowing the mineral salts to enter the root hair cell against concentration gradient. It's important to recognize that this does not apply so much for water. Water enters through the osmosis. Please be careful. So the next one will be relevant for water. Root hair cell has a thin cellular cell wall to provide a short diffusion distance for minerals to enter the cell and absorption of water by osmosis. Please be careful and not to use the term diffusion for water. Next, the vacuole, which has the cell set, is highly concentrated with it to provide a low water potential so that water can enter the cell by osmosis. So remember the 4 adaptations of the root hair cell. Now, coming to the next topic, diffusion and osmosis. What are the standard questions and answers? Number 1, describe what happened when a plant cell is placed in a solution with higher water potential. Water potential of solution outside cell is higher than that in cell set. And water molecules enter the cell through the partially permeable cell membrane by osmosis. The cell expands and becomes turgid. The cell wall will help to prevent the cell from bursting. Okay, very important to talk about the term turgid and not just leave it at expand. All right. How about what happened, or describe what happened when animal cell is placed in a solution with a higher water potential? The water potential of solution outside the animal cell is higher than the cytoplasm. So just like the description for plant cell, it is important to state the difference in the water potential. All right. And then the water molecules will then enter the cell through the partially permeable membrane by osmosis. And then the cell expands and bursts. Describe what happened when an animal cell is placed in a solution with lower water potential. The water potential of solution outside the cell will be lower than the cytoplasm. Water molecules leave the cell through partially permeable cell membrane by osmosis. We must write this down, partially permeable. And the cell shrinks and spikes appear on cell membrane. This is known as crenation. Number 7, describe what happened when a plant cell is placed in a solution with lower water potential. The water potential of solution outside the cell will be lower than the cell set. Water molecules will leave the cell through partially permeable cell membrane through osmosis. The cell decreases in size and becomes flaccid. Next, you may be tested on the functions of water. So these are the key points to take note. Number 1, water is a solvent for reactions. Give an example, photosynthesis. Water is a key component of cells, tissue fluids, and blood. Water transport dissolved substances, give example, digested products from the small intestine to other parts of the body, or waste products from cell to remove from the body. Number 4, water control the body temperature through evaporation or sweat to remove excess heat or remove latent heat to cool down the body. Next, food test is very important. It's important to remember the procedures to test for the various nutrients. How to test for reducing sugar? This is the complete description. First, place 2 cm³ of food sample into a test tube. Add the same volume of Benedict solution to the food sample. Shake the mixture and place the test tube into boiling water bath for 2 to 3 minutes. If the solution remains blue, no reducing sugar is present. If green precipitate form, traces of reducing sugar present. If yellow or orange precipitate form, moderate amount of reducing sugar present. If bright red precipitate form, there will be large amount of reducing sugar. So make sure you know what does the different color represent and remember the details in the testing of reducing sugar. Moving on to testing for fats. You realize that I did not talk about test for starch, not because it's not tested, because it's so easy, I will not waste time to mention it here. The test for fats is place food sample in test tube and add 2 cm³ of ethanol, shake the mixture thoroughly, add 2 cm³ of water to mixture. If cloudy white emulsion form, fat present. Solution remain clear, fat not present. To test for protein, this is the hardest for students remember because it involves more than one chemical where students tend to be confused. But it is very similar to reducing sugar, just that in the test for proteins, there's no need to place the test tube into boiling water bath. Add 2 cm³ of food sample in the test tube, add the same volume of sodium hydroxide and shake the mixture. Add 1% copper 2 sulfate solution drop by drop to the mixture and allow the mixture to stand for a few minutes. If protein is present, there will be violet colored solution form. If protein is absent, the solution will remain blue, which is the color of the copper 2 sulfate solution. How enzyme work using lock and key hypothesis? This question appears very frequently in the N-level examination paper. I would say at least 2 times for the past 10 years, at least. So let's go through this since it's quite important. Enzyme is the lock and substrate is the key. So students need to mention which is the lock and key first. Then students need to say substrate with 3D shape complementary, must say that point, to the active site of enzyme can bind and feed it to form enzyme substrate complex. Chemical reaction to form products and products are no longer complementary to active site and lead. This is important. You need to mention that the product is no longer complementary and therefore leave the active site, which make the active site available for the next substrate molecule to bind to it. So this is the description to explain how enzyme work using the lock and key hypothesis. Right? Moving on to the effect of temperature on the rate of enzyme activity. So we need to mention what happened from low temperature to optimum temperature and all the way to when it is denatured at high temperature. So this is the entire description. However, it depends on the question. Sometimes in the question, they may only require students or candidates to describe the effect by temperature before optimum temperature or after optimum temperature. So be careful, read the question carefully. If not, you'll be penalized. So at low temperature, enzyme is less active. As temperature rises to optimum temperature, the activity decreases because of increased rate of reaction. That will be sufficient. At optimum temperature, enzyme is most active with highest rate of reaction. Beyond the optimum temperature, the enzyme will start to denature and the enzyme activity decreases. It haven't completely denatured. There is still activity, but lesser. At very high temperature, when the enzyme completely denature, you will expect to see that the rate of reaction becomes zero and the activity reaches zero. Only when it completely denature, meaning the active site is totally deformed, distorted, and no longer can bind to any enzyme substrate. How about pH? At low pH, enzyme is inactive as the enzyme is denatured. When pH increases, the enzyme activity increases. At the optimum pH, activity of the enzyme become highest, and at high pH, enzyme is inactive because the enzyme is denatured. So some enzyme work best at acidic environment, some enzyme at alkaline, some enzyme work best at neutral. So do know the optimum condition for some typical enzyme. For example, pepsin found in the stomach is optimum at pH 2, while other enzymes like amylase is, if you talk about salivary amylase, it will be optimum at neutral condition, pH 7. If it is lipase, then it will be slightly alkaline condition. Describe the digestion of starch in alimentary canal. So you need to mention that, oh, in the mouth, there is chewing by the teeth to break up large pieces of food into smaller pieces. Digestion can mean physical and chemical. So if they mention digestion, then you have to mention both. If they only mention chemical, then you just specify the enzyme. If they mention physical, it's just the activity by the teeth in the mouth. So the starch is chemically digested by salivary amylase from where? In the saliva into maltose. The remaining starch is digested in the small intestine by the pancreatic amylase, by the pancreatic juice into maltose also. And finally, maltose is digested to glucose by maltase found in intestinal juice at the small intestine. Students have some problems recalling the enzyme, substrate, and the product. So as I presented in my previous video, it will be good that students come up with a table or refer to the table in the textbook to remember the specific enzyme, substrate, and product for the digestive process. Describe digestion of proteins in the alimentary canal. Protein is digested in the stomach by pepsin found in gastric juice to form polypeptide under acidic conditions. Remaining protein will be digested by trypsin found in pancreatic juice in small intestine to form polypeptide. And the polypeptide will be digested by the peptidase found in intestinal juice found in small intestine to form amino acids. And now we have the fats. For fats, you must mention the emulsification. All right. And the purpose of emulsification by bowel. And then you must say that the fat that is emulsified is digested by the lipase in both pancreatic and the intestinal juice to form what product. So it's important to recognize that you have to mention the substrate, enzyme, and the product in the digestion process. Let's move on to absorption. So after digestion, we have absorption. The inner surface is folded with many villi, with each villus covered by epithelium, which is one cell thick to provide short diffusion distance for substances to pass through. Okay, so this is one of the adaptation of the small intestine. We have villi, and on the villi, you have microvilli even. Okay, but it's more important to say that it's very thin and must say one cell thick, and it provides short diffusion distance. Small intestine is long and coiled to provide sufficient time for absorption of nutrients. Each villus contains many blood capillaries to help transport absorbed nutrients away to maintain the concentration gradient. Now, for functions of liver, it's not just stating. So you need to be specific in like, what does it do? What does it convert? So liver has 4 functions. You need to remember all the 4, it appeared before. Liver produce bile for emulsification of fats. Number 2, liver helps to maintain the concentration of glucose in the blood by converting excess glucose to glycogen and stored in liver, and will convert it back to glucose when the blood lacks of glucose. Number 3, liver deaminates excess amino acids, converting it to urea. Very important, must say this, deaminates excess amino acids. And number 4, liver break down the alcohol into carbohydrates used in respiration to release energy for cellular activities. Okay, move on to plants nutrition. Now, how does atmospheric carbon dioxide reach the cells in the leaf? Number 1, in daylight, the rate of photosynthesis is faster than respiration. Carbon dioxide is rapidly used up during photosynthesis. Carbon dioxide concentration in the leaf becomes lower than atmosphere. And then carbon dioxide from the atmosphere will diffuse through the stomata into the intercellular airspace in the leaf down concentration gradient. Carbon dioxide will then dissolve in the film of moisture on the surface of the mesosphere cells, and dissolved carbon dioxide will diffuse into the cell for photosynthesis. These are the adaptations of the leaf for photosynthesis. I will not read out everything. I will just choose 3 adaptations for clarification. I will choose thin laminar to provide short diffusion distance, because this word short diffusion distance came out very frequently. So I think it'll be easier for students to remember together. Stomata is heavily mentioned in this topic because of its importance to allow the movement of gases and water vapor. Stomata will be present at the epidermal layer to allow the carbon dioxide to diffuse in and oxygen to diffuse out during the daytime. That is what happened during photosynthesis. And the next one is chloroplast. The chloroplast is found in mesosphere cell, both the pelacyte and the spongy, just that you have more in the pelacyte because it's nearer to the surface of the leaf where sunlight can be captured more easily. This helps to absorb and convert the light energy to chemical energy to make glucose. So all these points must be mentioned. Anything missing, right, they will actually minus the mark. Okay, this is very important and often neglected by students, the process of blood clotting. So for this topic on transport of blood in the circulatory system, I have many details about like the chambers of the heart, the various blood vessels. I'm not going to put it here because they are more like labeling and answers are very short and sweet. So they may ask questions for the topic regarding the components of blood. What are they? How do they look like? Label them, describe their functions and perhaps adaptations. The chambers of the heart as mentioned, the blood vessels, aorta, vena cava, blah, 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 the valve in the heart, the direction of the flow of deoxygenated and oxygenated blood. What is pulmonary artery, pulmonary vein, and talk about why artery has a thicker muscular wall, blah, blah, blah. All these are very important, but I'm not going to mention here because they do not constitute very long answer. So what I have here is a template of questions that are rather long with more marks. The other parts of this topic, right, the answers are quite straightforward. Yeah. And normally students have no problem remembering them, normally. Okay. So for blood clotting, this is one of the main problems. I'll talk about another one later. This is one of the questions that students cannot remember because it's not intuitive. It is something that they need to put in more effort to remember the detail. And for this type of question, right, let's be very honest, after the exam, they will not remember. Okay, if you have teachers who teach other subjects, but they used to be very good in bio, they won't remember this also because it's not something that they will use it often in their communication. But you still have to know it for the exam purpose, okay, to answer the questions. So when the skin is cut, this enzyme called thrombokinase is produced and it will convert your prothrombin to thrombin. This thrombin will then convert the fibrinogen to insoluble tract of fibrin. So there is like 3 steps involved lah. Enzyme convert prothrombin to thrombin, thrombin convert fibrinogen to fibrin. And then this fibrin is the one that actually traps the blood cell to form the clot. So the clot is a fibrin in the 3rd step. And then the clot will seal the wound to prevent entry of microorganisms. This one, coronary heart disease is a common question because of the emphasis on health. So what are the key points to take note? Number 1, fatty substances like cholesterol and polyunsaturated fats, mentioned both, deposited on the inner surface of the coronary arteries. Okay, coronary arteries is the blood vessel that actually transport blood towards the heart muscle. Okay, so when the fatty substances deposited on the inner surface, it will block the movement of blood, which carries oxygen. So what happened is that this narrow the lumen of arteries and increase of blood pressure. So normally people with this condition will experience high blood pressure as well. So the affected artery will develop a rough inner surface to increase risk of blood clot. Yeah. And then the blood clot. So there's a blood clot formed in the artery, okay, due to the blockage, due to the inner surface that is rough. And then this blood clot will block the flow of blood and oxygen to heart muscle and causing heart attack. Okay, so you need to know the importance of sequence, the sequence of events. All right. Moving on to transport of materials in plants, starting with transpiration. How does it help to move the water up the plant? So number 1, the water will start to evaporate from the moisture on the surfaces of the mesophyll cells. Water vapor start to accumulate in the intercellular airspace, and then water vapor will diffuse out of the leaf through stomata via transpiration. All right. The water potential of cell will become lower than the xylem vessel because water diffuse out, and then water will pass from xylem vessel into mesophyll by osmosis. Remember, water moves by osmosis through the partially permeable cell membrane. And this generates transpiration pool to move the water up the xylem vessel from the roots to the leaf. Okay. Okay. Now, why transpiration is important? When water evaporates from the leaf, it helps to cool the plant to remove latent heat. Transpiration pool also causes the water and ions absorbed by roots to be transported to the leaf so that the water can be used during photosynthesis. Describe what happened to lactic acid after you exercise and why the level of oxygen intake remains high even after you stop the exercising. That's what it means. After intensive exercise, the breathing and heart rate will continue to remain high so that carbon dioxide can be removed faster and oxygen can be supplied faster to repay the oxygen debt. This ensures more oxygen and lactic acid transport to the liver faster for oxygen to convert lactic acid to glucose in the liver. Okay, so talk about all these points. Okay, adaptation of the alveoli, I will just talk about any two. For example, numerous alveoli to increase surface area, as well as one cell thick wall to provide short diffusion distance. You appear again. Okay, it depends on the question. So if in the question, right, they only show one alveoli, it's wrong to say numerous alveoli because from the diagram, they never show many alveoli. So it depends on the question. So if they say from the diagram, then you can say one cell thick, or if they show the moisture, you can say that. Or if they show the blood capillary, you can talk about the blood capillary. But for blood capillary, it's a bit lengthy. You must talk about what does this blood capillary do? What does it transport away? What does it transport towards the alveolus? and how does it maintain the concentration gradient. Or by the way, it's important to be careful with plural and singular. They will penalize students based on that. So if you say one alveolus singular, but there are more than one, is alveoli, okay, something like that. As for the properties of chemicals, I will not go through everything, but I have a sense that there will be a question on that this year because it never appeared for past 2 or 3 years. So do bear in mind the chemicals and what do they do. Okay, the easiest for students to remember is carbon monoxide because this chemical is also found in the chemistry syllabus. So it's easy for students to relate. The other one that students like is nicotine because they know what is adrenaline and the impact of it, and it's not too long to remember. Nicotine should be okay. So at least remember like nicotine, this one, carbon monoxide, this one, tau, what does it cause? It causes cancer, but you must mention uncontrolled cell division first before you say lung cancer. This one is the next one, chronic bronchitis and emphysema. Please know how to describe the sign. Okay, so for chronic bronchitis, must mention there will be excess mucus in the epithelium lining the air passages. So there will be excess mucus. Okay, the cilia on the epithelium become paralyzed. So the dust particles are trapped in the mucus, cannot be removed. So you will feel there's something stuck there in your air passage because it becomes blocked, making your breathing difficult. So you will cough frequently to clear the passage to help push out this mucus with the dust particle trapped there. Okay, so chronic bronchitis will paralyze your cilia so that the dust particles are trapped in the mucus, and then it will block your air passage, making your breathing difficult, and the person will cough to remove it so that he can breathe smoothly. So if a person moves constantly, he may have chronic bronchitis. As for emphysema, it's totally different. Emphysema may result from chronic bronchitis because if you need to cough frequently, right, the coughing will break the wall between alveoli. So chronic bronchitis affects the epithelium lining the air passage, but emphysema is related to the walls between the alveoli. So when the coughing breaks the wall between alveoli, it will cause the alveoli to become enlarged and inflate your lungs with air. This will decrease the surface area for gas exchange. Don't say volume ratio, just say surface area here. Okay, breathing will become difficult and the person suffer from breathlessness. Number 30 is the worst in terms of remembering. Okay, fertilization in plants. Students normally are very good with the label of the parts, but not very sure about this. So after pollination, sugary fluid on stigma stimulates pollen grain to germinate. A pollen tube grows out from each pollen grain containing 2 male gametes, must say 2 male gametes. Pollen tube grows down, style into ovary, enter ovule through microfile. And then pollen tube releases the 2 male gametes into ovule to start fertilization, where nucleus of 1 male gamete fuses with nucleus of female gamete to form the zygote. Okay, that's it. So you must have all this detail. What happened after pollination? There will be some germination of the pollen grain. That out of it grow the pollen tube with 2 male gametes. Pollen tube grow down from the style into the ovary, enter the ovule, my same microfile, and then the 2 male gametes release. Then there's fusion between the nucleus of the male and the female gamete to form zygote. That's it. So I hope this will help you. Thank you.